Dear students, welcome to the ninth session of Molecular Basis of Inheritance. Let's look into DNA fingerprinting. 99.9% of base sequence among humans is the same. Assuming human genome as 3 into 10 raised to 9 base pairs, in how many base sequences would there be differences? It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repetitive DNA because in these sequences a small stretch of DNA is repeated many times. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and other small peaks are referred to as satellite DNA. Depending on base composition, length of segment, and number of repetitive units, the satellite DNA is classified into many categories such as microsatellites, mini satellites, etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue from an individual show the same degree of polymorphism, they become very useful identification tool in forensic application. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity test in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism is the variation at genetic level which arises due to mutations. New mutations may arise in an individual either in somatic cells or in the germ cells. If a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, it can spread to the other members of population through sexual reproduction. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant at a locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence would be higher as mutation in these sequences may not have any immediate effect or impact in an individual's reproductive ability. These mutations keep on accumulating generation after generation and form one of the bases of variability or polymorphism. There is a variety of different types of polymorphisms ranging from single nucleotide change to very large scale changes. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. He used a satellite DNA as probe that shows very high degree of polymorphism. It was called as variable number of tandem repeats or VNTR. The technique as used earlier involved southern blot hybridization using radio labeled VNTR as a probe. It included isolation of DNA, digestion of DNA by restriction endonucleases, separation of DNA fragments by electrophoresis, transferring of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, hybridization using labeled VNTR probe, and detection of hybridized DNA fragments by autoradiography. The VNTR belongs to a class of satellite DNA referred to as mini-satellite. A small DNA sequence is arranged randomly in many copy numbers. The copy numbers vary from chromosome to chromosome in an individual. The numbers of repeats show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, the autoradiogram give many bands of different sizes. These bands give a characteristic pattern for an individual DNA. It differs from individual to individual in a population except in the case of monozygotic or identical twins. The sensitivity of technique has been increased by use of polymerase chain reaction. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider applications such as in determining population genetic diversities. Currently, many different probes are used to generate DNA fingerprints. Watch this video to understand more about DNA fingerprinting.
What is DNA fingerprinting or DNA profiling? Leicester University geneticist Alec Jeffreys developed a technique called DNA fingerprinting in 1985. It allows DNA samples from different people to be compared to look for similarities and differences. It's used for solving crimes and can also confirm if people are related to each other, like in paternity testing. Any two people in the world have 99.9% of their DNA the same, so this process analyzes the differences in the remaining 0.1%. This modern technology is called DNA profiling. It's a very sensitive technique which only needs a few skin cells, a hair root, or a tiny amount of blood or saliva. There are sections, or loci, of chromosomes, where instead of a gene consisting of a long sequence of bases, there are much shorter sequences of 3, 4, or 5 bases that are repeated many times. For example, these repeated sequences are called short tandem repeats, or STR. At these places on the chromosomes where we find these STRs, there are areas that vary in number of repeats. DNA profiling only looks at these STRs. A cell sample is collected. This could be from some blood at a crime scene, or a swab from the inside of someone's cheek, for example. The DNA is then extracted from the sample. Many copies of this DNA may be made using the polymerase chain reaction, or PCR. Special enzymes called restriction endonucleases are used to cut the DNA up into different size pieces. The DNA samples are then put into wells in a special gel called agarose for the process of gel electrophoresis, which separates the DNA fragments by size. The pattern is then transferred to a nylon sheet in southern blotting. And finally, the lines produced by the DNA samples from different people are compared. So let's have a go at using DNA profiles to solve some mysteries. An item was stolen in a burglary. A drop of blood was left behind by the thief. Samples of DNA were taken from 4 suspects and compared to the sample left at the crime scene. Which suspect is guilty? Here's a clue. Look for the one that is most similar to the one from the crime scene. Pause the video while you decide. Suspect 3 is guilty. Can you see how the pattern of bands matches in Suspect 3 and the crime scene? Their DNA is the same. Let's try another. A soldier has been killed in an explosion and has lost his dog tags that identify him. 3 soldiers are missing from their unit, so the army asks the 3 sets of parents for a DNA sample so that they might compare it to the soldier's DNA and therefore make an identification. Remember, the soldier will only share half of his DNA with each parent. So which set of parents is the soldier the son of? Pause the video while you decide. Parents C and D. As you can see, he inherited the first band from parent D and the second, third and fourth from parent C, and so on. So now you understand how DNA profiles or fingerprints are made and seen some examples of where they can be used. You can even interpret a DNA profile.
In today's session, you have learned about DNA fingerprinting and its uses. With this, we come to the end of the chapter Molecular Basis of Inheritance. Thank you and have a nice day.